In the first part of my talk, I will introduce the paradox of robustness. In essence, the better a system becomes at robustly protecting against perturbations, the more its components tend to decay in performance. So increasing robustness at the system level associates with a decay in component performance. And I'll develop that idea in the first part. In the second part, I'll relate the paradox of robustness to a variety of other ideas to build a broader conceptual framework. And in the third part, I'll use that broad conceptual framework to address a variety of interesting puzzles, for example, evolutionary aspects of development and the architecture of organismal design. So let's turn to the first part of the talk, where I'm going to develop the paradox of robustness by telling you two stories. And the first story is about cancer. And people generally say that cancer is a multi-stage process. And what they mean by that is that our bodies have multiple protections that protect us against disease. So for example, we have DNA repair in our cells, and that keeps DNA damage from accumulating. And often in cancer, DNA repair breaks down, and cancer cells accumulate a lot of DNA damage. So the breakdown of DNA repair is often thought of as one stage, potentially, in cancer progression. A second set of protections have to do with the cell cycle. We have many checkpoints on our cell cycle, so our cells do not just run through the cell cycle continuously and divide continuously, but instead there are checkpoints that prevent the cell cycle from continuing under most circumstances, and the cell cycle only continues under particular circumstances where those stops on the cell cycle are released. But in cancer progression, those checkpoints tend to break down or get knocked out. And so eventually the cell cycle runs continuously and cells divide more quickly. Another common protection against cancer is what's called programmed cell death or apoptosis. Our cells are designed so that if they're damaged or not behaving appropriately, this induces a cell suicide program. Again, programmed cell death or apoptosis. And often in cancer, apoptosis breaks down, and that allows the cells to divide and behave in ways that normally would have caused the cells to kill themselves, but instead the cells can survive and form a tumor. So we have these various protections against cancer, and people generally agree that cancer is some sort of multi-stage process. There's a lot of argument about what the stages are and how we should interpret the multi-stage process, but the idea of multiple protections is fairly widely accepted. And 20 years ago, in about 2003, when I began working on cancer, I was interested in evolutionary aspects of cancer. And one of the things I was thinking about was the fact that in different tissues in our body, we have different numbers of protections. And for the same tissue in our body compared to other animals, there are different numbers of protections, often depending on how long the organism lives. So it seems like there's evolutionary lability in the number of protections that exist for particular tissues. And so I became interested in thinking about that. For example, what happens evolutionarily if a new protection gets added to protect against disease? Well, in the first instance, that could be advantageous, right? You get another protection. And so what would have led to disease now is the disease is prevented because there's another barrier that must be knocked out before a cancer can spread. So that extra robustness mechanism or protective mechanism protects against disease and may be evolutionarily favored, at least at first. But then what happens if you add another protection? The existing protections, the selective pressure acting on those is now weaker because, for example, if apoptosis breaks down, but you have another protection downstream that protects against disease, the breakdown of apoptosis is not quite so severe in terms of damaging the organism. So the extra protection weakens the selective pressure acting on the previously existing protections that were there. And as evolutionary pressure weakens on those protections, then we might expect more genetic variation to accumulate. I've shown with a variety of analytical models that we get an increase in the heritability of disease which may explain the puzzle of why the heritability of disease is often so high. And also those components that are now partially protected by this later protective mechanism, the earlier components, they may decay in performance and become sloppier. Maybe they'll decay to a cheaper, lower performing, sloppier component because stochasticity or a lower cost, lower performance of such components is no longer so severe and so strongly selected against. So it may be advantageous to the organism to invest less in those earlier stages of protection. So we add a protective mechanism, and the existing mechanisms tend to relax and decay a bit. And this leads to a certain irreversibility because we've added a protective mechanism, the other components have decayed. Now if we pull off that additional protective mechanism, the system is going to perform very poorly because the existing components have decayed in performance. So we have a certain irreversibility. People sometimes say this is like an evolutionary ratchet. You might add a protective mechanism, you get a decay in components, and you cannot go backwards. So then you might add another protective mechanism, and you might get layering of protective mechanisms and decay of the components below it. So when I was working on that, I realized that this description or idea applies not just to cancer, but it applies to any aspect of robust system design, error correction, repair, homeostasis, canalization. Many things that our bodies do have properties that are like robustness. And so maybe they're associated with decay as they evolve. And I thought, well, this could be really quite a widespread evolutionary principle that would help us to understand a lot about the architecture of organismal design. But it's challenging to study empirically because one would have to be able to see the origin of a new protective mechanism and then the decay of the existing components as this additional protective mechanism is added to the system. And it's very hard to get that kind of resolution in empirical observations. So I continued to search for examples that might bolster this idea. And the best example I could come up with actually comes from engineering. So that will be my second story to introduce the paradox of robustness. And this has to do with computer hard drives and data storage. So about 30 or 35 years ago, computer hard drives were very expensive. A little hard drive, tiny hard drive, by modern standards, cost thousands of dollars. And they were very expensive in part because they had to be engineered to very high tolerances and low failure rates. Because if you're storing your data on a hard drive and the hard drive fails, that's catastrophic. You lose all of your data. Or if errors are creeping in on the data that are stored, that's also catastrophic because then you cannot trust the information that you stored on the hard drive. And so over time, people have developed various strategies for dealing with this problem of hard drive failure. And essentially all mid-range and high-end computing systems today use what are called RAID arrays, or redundant arrays of independent disks. In the simplest case, redundancy is very effective. You have a hard drive, you store your data on this hard drive. But if you add a second hard drive, a redundant hard drive, then when you save a file on your computer and the data come to be stored, they can make a copy of the data on the first drive and a copy of the data on the second drive. And so if one of the hard drives fails, you're okay because you've got another functional hard drive. So redundancy is very effective, but it's also very expensive because it doubles your cost in terms of data storage. And it's slow because the data have to be copied twice onto the hard drives. So people have figured out cleverer ways of taking advantage of redundancy. So in essence, what essentially many computing systems do today is they use redundant array of disks. But instead of just copying the data on each disk, they have a little mini computer that sits above the array of disks. That's called the RAID array controller. So this is like a computer board that's a little mini computer. And when you save a file on your computer and the data come in to be stored, instead of coming into the hard drives directly, it goes to the RAID array controller. And this little computer breaks up the data into a bunch of little chunks and spreads those data chunks across the array of disks. But it does it in a clever way. It's not just a copy of the data on the first disk and a copy on the second disk and so on, because that's very expensive. but it breaks up the data chunks and spreads the data chunks across the array in a sort of complicated algorithmic way that maximizes redundancy and safety and keeps performance up and reduces the number of disks you need. And in fact, you can tune the algorithm. You can make it so that there's a lot of redundancy, and that increases your safety against failure, but it means you need more hard drives to store the same amount of data, and so it's expensive and it's slow. Or you could use a bit less redundancy. That's a little bit less safe, but you need fewer hard drives and you have higher performance. So that's advantageous. So you can tune the algorithm, and people actually do tune these algorithms. But what's interesting to us is that once you have this RAID array controller system with redundant disks, then people have these systems and they're running. And if one of the hard drives fails, you just pull out the failed hard drive, you stick in a brand new hard drive without ever turning off the system, and the system knows how to rebuild itself because it has copies of the data on the other disks, and so it rebuilds itself so that no data are lost, and it's back to its initial state. And in addition, if errors creep into the data, you have redundancy and error correction, so you can protect against errors being produced on the data storage medium. So this is a very robust system. And people have recognized that because a hard drive failure is not really such a big deal anymore, because you have this redundancy in a system for rebuilding itself and repairing itself, that you don't want to use expensive hard drives, because if a hard drive fails, it's not that big a deal. You just pull it out and stick a new hard drive in. And so I've seen online analyses and white papers written by companies that do huge amounts of data storage, where they talk about what's the best hard drive to use in this kind of system. And they look at a variety of available hard drives that have different costs and different reliabilities. And they actually favored choosing a cheaper disk that has lower reliability rather than a more expensive disk with high reliability. Because economically, it's advantageous to use cheaper, lower performing, disks more susceptible to failure, because failure is not a big deal, and so you want to save money at this level by not using very expensive disks and let the system robustness take care of the problem. So we then get what I've described. You have a higher level robustness, and you get a decay in performance of the components to cheaper, lower performing state. So I've described two different stories. I've told you about cancer, and I've told you about these RAID array computer hard drives, describing how the paradox of robustness could potentially work. And I think this, as I said, I think this is a very powerful idea that could be widely important. But now what I want to do is move to the second part of the talk, where I relate the paradox of robustness to various related concepts and ideas to build a broader conceptual framework. So we'll now move to this second part. And first, I'll just mention a few ideas very briefly. There was one idea developed by Susan Lindquist in the 1990s, so this was independently developed by her. And I'll talk about her idea in more detail in a minute. So I'll just briefly say that she was working on a system where there was a higher level factor within, this is now back to biology, higher level factor within cellular biology that protected against perturbation at the lower level. And she recognized that once you have this higher level of protection, that the lower level, some of the lower level perturbations would become selectively neutral in the presence of this protective mechanism, and that would allow the accumulation of genetic variation. And that has various evolutionary consequences. Again, I'll come back to this. A second idea is known as neutral networks. And so once again, if you have some higher level protection, or the system is designed in a way that a variety of epistatic interactions that form some sort of network of interactions at the cellular level is relatively neutral, so that you can get different genetic and genomic components, but they interact in a more or less similar way functionally, that you can get drift along some sort of neutral network in a flat part of the fitness landscape. And this can lead to novel evolutionary consequences as you drift along through this flat evolutionary landscape. It's going to be very important for understanding evolution of novelty. And so this idea has been widely developed by many people including, for example, Andreas Wagner, but many other people have contributed to this idea. And the third idea that I want to mention briefly is fitness landscape theory in general, because in essence, the paradox of robustness is about altering fitness landscapes. And fitness landscape theory has been developed quite generally, particularly in the study of viruses, because in viral biology, there's a lot of data about genetic sequences, phenotype, and fitness. And there's also been a lot of advanced mathematical development. And I think in the long run, it will be important to bring in the advances in fitness landscape theory to understanding the paradox of robustness. But at present, that has not yet been done. But the main idea that I want to develop today is an idea called constructive neutral evolution. And this is an idea which is very similar to the paradox of robustness. And I only learned about it a couple of months ago. And so actually, one of the main purposes of today's talk is for me to work out what's the relationship between constructive neutral evolution and the paradox of robustness. And as you'll see, I think that there's some real advantages in thinking about the ways in which these ideas are complementary. They're quite similar, but complementary. So let me tell you about constructive neutral evolution. And this idea was started originally about 30 years ago, which predated my own work by about 10 years. And it was developed mostly from a group of people in Dalhousie in Canada, working with Ford Doolittle, either directly or indirectly. And the first people to publish on this were Michael Gray, Arlen Stoltzfus, and a variety of other people. And Ford Doolittle worked with them, sometimes directly publishing with them and sometimes indirectly. And later, also in this group with Ford Doolittle, Tyler Brunet has worked with the group to develop these ideas further. And many other people have contributed to these ideas. So it's developed into quite a broad field of study. And as I said, I've just learned about it, and so I'm still working my way through the idea and the way it relates to some other ideas. So let me introduce the constructive neutral evolution point of view with a couple of examples. The first example they developed in their own work was on RNA editing. So we have typically DNA makes RNA makes protein. So that's the standard view. But it turns out that in some organisms, under certain circumstances, DNA makes RNA, so you have an RNA sequence. But then some enzymes edit the RNA sequence and transform the RNA sequence into a different sequence, and then that sequence makes protein. So you have DNA makes RNA, RNA makes a new RNA sequence, and then you get a protein. So it's a bit more complicated. And so how does that work? Well, if you think about an RNA sequence, then at some point there was an enzyme that came along that changed some of the bases in the RNA sequence to different bases, different nucleotides, and that changed the amino acid sequence. Maybe initially that was just random, or maybe it was selectively favored because of the changes were advantageous. It doesn't really matter. It could happen either way. And so you might imagine, for example, that there's an enzyme which changes C nucleotides to U nucleotides in the RNA sequence, where U in an RNA is the same as T in DNA. RNA uses U, DNA uses T. They're essentially the same thing. So you have an enzyme that changes C to U. So DNA makes RNA, you have an RNA sequence. In the RNA sequence, if there's a U, it stays U. If there's a C, it changes into U. So C goes to U and U goes to U in the RNA sequence before it then is made into a protein. That means at the DNA level, if the DNA nucleotide codes for a U in the RNA, then it stays U. If the DNA nucleotide at a particular position codes for C, it becomes U. So either of those two nucleotides then end up coding for U. And so RNA editing imposes a selective neutrality at the DNA level, whereas previously those alternative nucleotides at the DNA level coded often for different amino acids. Now it's selectively neutral because the two different nucleotides are both making U. So we've got neutrality at the DNA level created by this RNA editing process. That means there's going to be neutral drift at the DNA level, where these alternative nucleotides are going to drift in frequency. And now if we took away the RNA editing, we would have alternative nucleotides at the DNA level, but they would code for different nucleotides in the RNA. And they might code for different amino acids, so they're no longer selectively neutral in the absence of RNA editing. So we get a system where you cannot go back. Once you've added the RNA editing and you have the neutral drift at the DNA level, then this becomes an irreversible ratchet. And this is exactly the way people who work on this constructive neutral evolution idea talk about it. You get a complexification of the genomic architecture, an irreversible complexification. And they were really interested in the fact that eukaryotic genomes seem to be ridiculously complex. If you study eukaryotic genomes, they do all sorts of crazy things. And their argument is that those crazy things probably mostly are not adaptive, but they're the consequence of having some sort of buffering, robustness, suppression mechanism that suppresses the consequences of variation in some other part of the genome, like RNA editing. You get a neutral drift, and now you have irreversibility. And so you've added another step in the processing of information. Things are more complex, and you can't go back. And I think I'm fully supportive of this idea, and I think it's very similar to the paradox of robustness. We'll talk about that in a minute. But let me give you a second example, and this goes back to Susan Lindquist's idea that I mentioned a minute ago. Working independently in the 1990s, Susan Lindquist was working on heat shock proteins in Drosophila, for example, HSP90, which is a heat shock protein. And one of the things she found is that this heat shock protein acts as what's called a chaperone that helps proteins fold correctly. So proteins start out as an amino acid sequence, and then they have to fold up into a three-dimensional conformation to be functional. And sometimes they don't fold correctly. And these chaperones, these heat shock proteins, these chaperones, help primary amino acid sequences fold into proper functional forms. So that's advantageous. They add some robustness to the system, or buffering. And Lindquist recognized that once you add this buffering that helps proteins fold correctly, that you might get some mutations in the primary amino acid sequence that in the absence of the chaperone would cause protein misfolding, and so would be strongly deleterious. But in the presence of the chaperone, the mutated amino acid sequence folds correctly and is essentially functionally equivalent. So what you've done is added a suppressive mechanism, a buffering or robustness mechanism, that now has rendered relatively neutral amino acid substitutions that were previously would have been disadvantageous. And so Lindquist then said, well, that's going to lead to the accumulation of genetic variation by this buffering. And her interest was that although that genetic variation would be neutral in the current environment, if the environment shifts, some of that variation might become selectively advantageous. And so this pool of genetic variation that gets created acts as a storage of genetic variation that can increase the ability of the organism to evolve to new challenges in the future. So she was interested in the evolvability advantage that arises from this process. Constructive neutral evolution would look at the same thing slightly differently, same process, but they would say, okay, you get a chaperone that makes different amino acid substitutions nearly neutral. That means you're going to get an accumulation of genetic variation. Now you've got an irreversible complexification in the way in which information is processed, because initially the organism did not need the chaperone, but once you put the chaperone there and you get the relaxation and drift at the lower level, you cannot go back. And so this becomes a necessary component of the way in which information is processed. You've got an increase in complexity. You've constructed greater complexity from neutrality. So those two ideas, RNA editing and the chaperone idea, give us a sense of the concept of constructive neutral evolution. In my own work, I like to say that genomes are overwired. If we look at the factors that control the expression of a particular gene in the genome, there's transcription factors that bind near the primary DNA sequence, and those transcription factors can enhance or repress the expression of a gene. There's enhancers and suppressors that act at a distance across the genome. There's histones, which influence the way in which the DNA is wound up, and that can affect the ability of the DNA to be expressed and form RNA transcripts. There are various different kinds of RNA that act at a distance that influence the expression of a gene at many different stages in the process, and so on. There's just so many different ways that affect gene expression. So I would say this is overwired. We're getting a lot of connections and control factors that influence gene expression. And I say this is overwired because an engineer making a system to control gene expression would never make it so complicated and so densely wired. It would make something simpler and easier to understand and control. So we see this great complexity in genomes. Genomes are overwired. So that's basically constructive neutral evolution and the ideas of the way in which we get complexification at the genome level particularly. And so what I want to do now is put together the idea of the paradox of robustness and constructive neutral evolution and make the argument that although they're very close conceptually, that they really emphasize quite different things. And when we bring these two ideas together, we get a much more powerful conceptual framework that can really help us to connect up a lot of things that were previously unconnected. And so that's what I'm going to do in the third part of the talk. And I'm going to start this third part by just telling you about a couple of observations that have been made about organismal design that have nothing to do directly with the paradox of robustness and constructive and neutral evolution. And then I'm going to tie things together and show how we can develop a broader framework. So the first idea I'm going to tell you about is what's called the hourglass model of development. And the hourglass model of development comes from people that study the development of closely related species. And they look at how the developmental process diverges between a set of closely related species. And one of the observations that often arises is that the earliest stages of development tend to diverge fairly rapidly from each other. So you get broad divergence in the earliest stages of development. The intermediate stages of development often are highly conserved and do not diverge between species that are closely related. And the later stages of development that make the adult morphological form, those diverge fairly rapidly. So you get an hourglass, broad divergence at the earliest stages, coming together in kind of a shared, constrained set of developmental processes in the intermediate stages of development, and then a divergence in the later stages of development. And this has been discussed widely in literature for a long time. Some of the interpretations are quite controversial, but the basic notion of the hourglass is often supported by molecular studies. For example, there was a study published just a couple of months ago on C. elegans, which is a little worm-like creature. And they looked at a variety of closely related species, and what they found is that the proteins that influence the earliest stages of development in C. elegans tend to evolve fairly rapidly and diverge fairly rapidly. Those proteins that influence intermediate stages of development tend to evolve quite slowly. So you have a conserved middle stage. And the proteins that affect the latest stages of development diverge fairly rapidly. And in this study, they related those observations to the hourglass model of development. So this is something that arises and gets discussed in the literature. So for now, just hold that hourglass model of development, because I want to talk about a second hourglass that gets discussed. And this is going to originate from engineering, where people talk about the basic properties of complex, robust systems. And in particular, this comes from the work of John Doyle. And John Doyle has done perhaps as much as anybody to develop modern, robust control theory. This is the theory that's used to build airplanes, to build automobiles, to build internet protocols, is widely used throughout industry to build many of the things we use in daily life. They use what's called robust control theory, and Doyle developed a lot of the principles that are used. And Doyle's very interested in the general architecture of engineered systems, and he's also very interested in the architecture of biological systems. And so he's written a lot about complex, robust systems in both biology and engineering. And one of the things he emphasizes is an hourglass model that has the following properties. What Doyle says is that at the lowest level of some sort of complex engineered system, this would be at the physical level, how you actually build a system from the lowest level physical components that you're using to put together a system, sort of down in the electronic level or the low little building block levels, that these tend to be quite diverse. People build up hardware structures in a variety of ways. But as they come together to become more functional, and I'll give you an example in a minute, there tends to be a narrow set of protocols that allow higher level systems to talk to the low level hardware. So you get a little narrow waist, and then at the functional level, which might be the software level, you get a wide divergence of different functions. So you get this hourglass model of design. And this will make more sense as I give you some examples. One example that Doyle talks about are cell phones. He said that modern cell phones, different companies make cell phones, and they build the hardware in a variety of different ways. And they have quite a bit of flexibility because cell phones are basically little computers. And computers have to satisfy at the hardware level just some basic constraints. They have to be able to store data, and then they have to be able to retrieve that data at a later time. They have to be able to build basic digital logic so that the higher level computing system that sits above it can write computer programs. So they just have to provide some basic services. And there's a lot of different ways of building at the hardware level these basic services. And so cell phone companies do this in a variety of ways, and it would even be possible to do it in even a wider diversity of ways that are not used for practical reasons. There's a lot of different ways of building such hardware that provides these basic services. But all of these hardware designs tend to use a common set of protocols. And those common set of protocols are like an operating system. So it's like Windows or the Mac OS. And in fact, they use the Linux operating system. And at the lowest level of all operating systems is what's called the kernel. And the kernel sits between the hardware and the software and forms protocols. So when you write a software program and you tell the hardware, store some data, I'm going to need it later, just store it for later. The software doesn't know anything about how that's done at the hardware level. It just tells the operating system, here's some data, store it. And then the kernel gets some calls and it says, oh, I need to store some data. And it knows what's going on at the hardware level. And so it stores the data down at the hardware level. Then later, the software says, I need that data back, give it to me. And then the kernel says, okay, I know where the data are. I'm going to find the data. I'm going to bring it back and I'm going to let you have it. So these protocols sit between the software and the hardware. And the protocols are highly conserved because that allows different hardware architectures to work with different software architectures because they all use this common set of protocols that go through this narrow waist. And so at the low level, you get a lot of diversity at the hardware level. You get a common set of protocols. And then you get diversity at the functional level, which could be software or other kinds of function. So you get an hourglass model that's a common design for robust, complex systems. And Doyle says that airplanes and automobiles and internet protocols and many other things have this basic hourglass design. This is a universal attribute of the design of complex, robust systems. And Doyle's also written quite a bit about biology. And he says the same thing is true of organismal design, that they have this hourglass tendency of design. For example, if you think about how we power our cells with ATP versus ADP. So the molecule ATP relative to the molecule ADP, the disequilibrium between those molecules is what powers essentially all of life. And so core metabolism is basically the process of building up an ATP-ADP disequilibrium that acts as a battery storage pack that's used to power life. And so if you think about it, basically almost all of life is using this ATP-ADP disequilibrium at its core for its power. That's from the earliest cells, archaean bacteria, on up to our bodies. They use a very similar set of protocols, you might say, biochemical reactions. But if you look at the earlier stages that feed into this protocol of power, organisms feed on a variety of different molecules, and they have a variety of biochemistries early in the biochemical cascade of metabolism. So you get quite a bit of diversity. But as that cascade works towards moving the molecules and their products towards the central core, where they're going to transform ADP into ATP to be the power source for cells, they use a common set of biochemical reactions that are highly conserved. There's some variation, but the conservation is fairly amazing across huge spans of time. So you get wide diversity in the metabolic biochemical level in terms of acquiring basic components to feed into core metabolism. Once you have an ATP-ADP disequilibrium, you can use that to power building up all sorts of complex biochemical and morphological structures. You can power behavior. You can power all the things that organisms do. And so you get a great diversity. So you have a very narrow central waist in the hourglass, right? Diversity at the bottom, a narrow waist in these common protocols, and a diversification of functions. The same thing is true for DNA makes RNA makes protein. Although there's slight variations of that across life, basically all of life uses that core set of protocols for transforming the digital information in the genome into the proteins that are the functional components of life that basically build the software and functional layer of life. So there's a lot of diversity in the hardware layer in terms of genomes and the way information is stored and transmitted and manipulated. But all of it flows eventually into the core protocol of DNA makes RNA makes protein, right? With some small variations, but basically it's that basic core. And then you get a diversification into software level. And Doyle talks about it this way. He says, okay, you get this narrow waist in the hourglass model of the architecture of organismal design. And I'll tell you a third and final story here, and then we'll tie things together. A couple of months ago, an article came out by a group at Tufts University in Mike Levin's lab, and they study developmental biology of planaria, which is a different kind of little worm-like creature. And they do a lot of empirical work, developmental biology work at the molecular level, studying this organism and a variety of closely related species. But they also do a lot of very creative theoretical work. In the article they published a couple of months ago was a theoretical study designed to try to understand how development evolves. And so they made some computer simulations where they had a genomic level that encoded for not phenotypes, but simple rules of development. So the idea was that the genome can make simple rules of development, and then those rules of development must interact with each other to make the final adult morphological form. And so they were curious how the system would evolve if there was some developmental challenge. What would be the relationship between the genomic level, the rules of development, and the way in which development proceeded to make the final organism? And one of the things they discovered was that fairly early on in the evolution, in the computer, the evolution of this system, was that the genome would tend to make a core set of low-level developmental rules. And then those low-level developmental rules would interact with each other to make the final form. And so they actually used the word hardware for the low level and software for the higher level. And they noted that the hardware is basically encoding for a small number of simple rules. And those simple rules are then interacting to make complex software, complex morphological forms. And so they essentially recreated this hourglass notion of development, where there's variation at the lowest level, and then you get a core set of protocols at some stage in development. And then at the highest level, you get diversification. And they called the lowest level hardware and the highest level software. And they had a core intermediate thing. And they also have recreated Doyle's hourglass model for robust complex systems, because they noted that at the hardware level, once you get this structure, that the genome hardware level tends to diverge fairly rapidly, and the software level is diverging, but the core set of protocols are relatively conserved. So this is like Doyle's hourglass model. But they didn't mention Doyle's work, and they didn't mention the hourglass model of development. And I find that very interesting because they've, in essence, recreated these prior ideas. And when people are recreating prior ideas, this is really a big hint that the time is right and people are on to the same set of ideas. And that we should pay attention to this. This is actually suggesting that the time is really ripe for a greater synthesis, bringing these things together. And so that's what I now want to do, is I want to suggest a little synthesis. Now, I've only been working on this. I've only known about constructive neutral evolution and thinking about the synthesis for about 6 weeks. So this is fairly new, but this is my first pass. Here's how I'm going to bring together constructive neutral evolution and the paradox of robustness. I'm going to say that constructive neutral evolution has always tended to focus on the genomic hardware level. They've worked on things like DNA, RNA editing, and various complexifications that happen in the genome, where there's a tendency for neutrality and drift. And the paradox of robustness came out of my studies of cancer. I was interested in cancer, in physiological homeostasis, in developmental canalization, in behavior, and really what's a software functional level. And at the software functional level, I think economics is going to be more important. For example, if you think of the computer hard drives, when you add a RAID-array controller, the computer hard drives don't drift neutrally. They still have to function. But they can function in a sloppier way and in a less costly way. So to me, at the functional level, you're going to tend to get a decay to lower performing, less costly, sloppier components by an economic marginal relaxation of costs and benefits, which is quite different from the neutrality that's discussed at the genomic level. And so if we bring these two ideas together, where the constructive neutral evolution view maybe dominates at the hardware level, and the software paradox of robustness view dominates at this higher functional level, that we now have a very broad framework that can cover a very wide variety of phenomena. And having done that, we can tie together with notions such as the hourglass model of development, where you have early levels of diversification, which are more like hardware genomic early stage levels that are just the building blocks, the hardware pieces, and later stages of diversification at a software level, and a common set of protocols which acts as a connection between the hardware and software levels. And the protocols act also as kind of a robustness mechanism, because you can get sloppiness at the hardware level as long as the common protocols are respected and obeyed. And you can get diversification up above. So the robustness notions fit into this hourglass notion of development, and they fit into Doyle's hardware versus software notion with an intermediate set of protocols for robust complex systems. So now we see many pieces fitting together with the constructive neutral evolution with a kind of dominant emphasis on the hardware level, the paradox of robustness with a more dominant influence at the software or functional level. Although the ideas are very similar, their different emphases really help us to see a much, much broader array of phenomena and see a unification. And so that was the main point of today's talk, was to propose this potential unification of ideas into a broader framework. And I could stop there, but I want to go on and give you one more connection, because the real strength of this kind of framework is the ability to bring in a variety of things that were seemingly unconnected and give us a common framework from which we can understand it and how things might be related. And that's really a conceptual advance. And you know you're onto something that's working when you start to see various things that seemed unrelated connecting up. So I want to give you one more example of how that might be true going forward. And this last example comes from machine learning and artificial intelligence. And one of the great breakthroughs in modern machine learning and artificial intelligence happened about 10, 15 years ago, when people started using deep, densely connected computational neural networks to solve problems. That's when people first started noticing that you could build systems that were extremely good at recognizing images and eventually in beating humans and playing chess. These are deep, densely connected neural networks, computational neural networks. And there was a real revolution when people started making these overwired networks. And I'll tell you what I mean by that in a second. Remember I said that genomes are overwired, that the number of connections that are involved in controlling gene expression is very high. Many factors influence it, gene expression. And I suggested that that overwiring may arise by the paradox of robustness. You get a higher level robustness mechanism that sits above a system, you get some decay in the components. You put another robustness mechanism on top of that and decay of components. And this is irreversible. So there's a natural tendency for deep, densely connected, layered architectures in the control of our cells. And it turns out that in artificial intelligence and machine learning, when you make deep, densely connected, layered architectures, that this revolutionized machine learning, and machine learning then could accomplish so much more than it ever did before. And this is interesting because when I was taught statistics, I was always taught if you want to make a model that's very good at making predictions, you should use the smallest number of parameters that you can. You should reduce the model as much as possible. And what we've discovered is that the opposite seems to be true for learning, that making highly over-parameterized models with many more degrees of freedom than are needed actually allows systems to learn very quickly. And so that's one of the great advances. And one of the puzzles in modern machine learning is that why is over-parameterization so successful in enhancing the rate of learning? And in evolution, we might ask, as we started to get deeply, densely connected, layered control structures in our genomes, maybe that unlocked a great evolutionary explosion. People have suggested that more or less, but I'm now tying this into the paradox of robustness and the overwiring and emphasizing the over-parameterization. And that's something that really has not been emphasized in biology. What I'm saying is that the over-parameterized nature of our control architectures may actually be a key aspect that allowed evolution to proceed very rapidly. And so by bringing together insights from machine learning and evolutionary biology and engineering and robust control, I think we're going to get a much broader understanding of the relationship between learning and evolution, which after all are very similar because evolution is just a particular type of learning. And we're going to begin to understand these things and link them up with these various hourglass models, paradox of robustness, constructive neutral evolution. I see these things coming together to form a really interesting and cohesive framework that can help us to understand many puzzles in organismal design. Thank you.